evening, all the aspirants present here. Here we have Dr. Saibal Moitra. He is a professor and senior consultant, allergy and immunology department of pulmonology at Apollo Multispeciality Hospital, Kolkata.
Good evening to all. So this is in continuation to the lecture of immunology. And I think in the last lecture, we discussed about the innate immunity. So if we just quickly recollect the innate immunity, what we had discussed so far is that there are two arms in the immune system, the immune response. One is the innate arm and other is the adaptive arm. So whenever our body encounters any threat in the form of, say, let's take is the any microorganisms. We know there's panoply of organisms all around us, and is always trying to get inside our system. So this immune system is always it is protecting us. There's in every second. Okay. So how it does? So one arm is that which is already there, and so means irrespective of whatever the type of the microorganisms it encounters, it delivers a certain set of the fixed pattern of the immune responses. So that is the innate arm. Innate arm, we have some structural part or the cellular component of the innate part, and we have the humoral component of the innate immune system, or the cellular component or the structural component. Structural component, first we say it is the mucous membrane, it is the skin, it is the epithelial barrier. Okay, these are all the structural part of the innate immune system. Nowadays, we also say that the microbiota, which is it is also a component of the innate immune system, because it is always interacting with our other components, that is the epithelium and other cells of the innate immune system, and it helps in modulating the immune response to a pathogenic microorganisms. So in the cellular component, we have, first of all, the sentinel cells, which is always guarding our body, and it is just beneath the epithelial layer. So the sentinel cells, mainly we talk about the three main types of the sentinel cells. One is the tissue resident macrophages, then the mast cells, and obviously the dendritic cells. And what these cells does, these cells all carry some innate immune receptors. These innate immune receptors are also called the pattern recognition receptors. Why? Because they identify certain set patterns in the microorganisms, and which are present only in the microorganisms, and they are not present, these molecular patterns are not present in us, and these molecular patterns are very important for the survival of the microorganisms. And what are these patterns? These pattern molecules. Pattern molecules, it could be in the bacteria. When you talk about it in a bacteria, it could be the peptidoglycans, it could be the teichoic acids, it could be the flagellins, and if we talk about the viruses, it is the nucleic acids. That is a single-stranded RNA, double-stranded RNA, and a double-stranded DNA. These are all the specific molecules which give the identity to a microorganisms. And this is molecular patterns. They're recognized by these innate immune receptors or the pattern recognition receptors. And so these are called the pathogen-associated molecular patterns, or PAMPs. Many a times we have the cells which is dying cells. They also exhibit certain molecules in the surfaces which are recognized by the innate immune cells, and they kill them. So they're also called that is the danger associated molecular patterns, or DAMs. So we have these PAMs and DAMs, whatever it is, recognized by the pattern recognition receptors. And we discussed that we have multiple sets of the pattern recognition receptors. The most common which we discussed, the archetypal PRRs, are the toll-like receptors. So toll-like receptors, it could be that is the surface toll-like receptors, or we can have the endosomal toll-like receptors, and like TLR1, TLR2, 3, 4, 5, and all these TLRs. And these TLRs, basically, they recognize the molecular patterns, particular molecular patterns. And when this TLR means attaches itself to the ligand is basically there. I would request everyone to switch off their microphone, otherwise it becomes very difficult to continue the thing. Sana, please ensure that everyone has switched off their microphone. or if there is a possibility, then you just switch off everyone's microphone. Okay.
Yes, sir. Sure.
Yes. And so what happens is that this when this is toll-like receptors or pattern recognition receptors, they engages with its ligands. The ligands are the PAMPs. They send some downstream signals in the cells, and these signals leads to the expression of the various molecules on the cells. And not only those molecules are expressed, it also leads on to the release of various cytokines in the cells. It causes activation of these innate immune cells else. So along this takes care of the microorganisms, and two main mechanisms of the innate immune mechanisms we have talked about. One is the inflammation, and another is the antiviral state. Okay, so and there are very various humoral components of the innate immune system as well. That is the most common proteins or the soluble components of the complement system. We have seen that there are three. There's one is a common pathway of the complement system, and the main component complement system is the complement protein 3, and these complement proteins are synthesized in the liver. And this complement 3, it gets cleaved into the 3A and the 3B, and this 3B, it then causes the cleavage of this complement protein 5 into 5A and 5B. Then the 5B, it then attaches to C6, 7, 8, and 9, and causes a causes a multimeric molecule which can go and insert into the cell membrane, because it is put amphiphobic, and then it causes the formation of pores in the cell membrane of the microorganisms, also known as membrane attack complexes, cause lysis of the cells. But apart from this common pathway, there are three separate pathways which leads on to the C3 activation of the cleavage. It could be because of the antigen antibody reaction, and which is known as the classical pathway, where IgM is basically important in activation of the classical pathway. Or we can talk about the innate pathway. And then we have the alternate pathway, where this is activated by the surface contact act with the microorganisms. So we have the lectin pathways, where these lectins, like the mannose binding lectins, they're important, and in activation another cleavage of the C3. So apart from that, we have other humoral components, like we have the pentraxins. One of the common pentraxins we talk about is the C-reactive proteins. We have the lectins. We have the natural antibodies of the IgM, which do not require the previous sensitization, but they can directly go and bind and destroy any microorganisms. And we have also discussed about the various other cellular components, that is the natural killer cells, and also all the components which are derived from the myeloid component of the hematopoietic stem cells, namely the neutrophils, the eosinophils, the basophils, that all form a part of the innate immune cellular components. Now, so once this is the innate immune system, it handles the microorganisms and able to kill it. It does its job with the due to inflammation. Another is the antiviral state, and it does its job. But if it's not able to do it, then what happens, that information is carried to the secondary lymphoid organs or the regional lymph nodes. And this is done by the antigen presenting cells. And one of the main antigen presenting cells that we talk about are the dendritic cells. So what this dendritic cell does, dendritic cell basically, dendritic cells, they're the antigen presenting cells. So here, if you can see that this is the pathogen, and this is then and pattern recognition receptor, maybe a TLR. Okay, and this is also a TLR. Okay, now it recognizes this pathogen. And because what happens is that what this is antigen presenting cell does, it just engulfs the pathogen, and it then destroys it by the various lysosomal enzymes, and cleaves it into smaller peptides. And these peptides are then presented with an MHC class 1 or an MHC class 2 protein on its surface. This is one thing it does. So the second thing, what it does is this is the binding of the pathogen with the TLR, the PAMP with the TLR. It causes the activation of the co-stimulatory molecules, which belongs mainly to the B7 family. at the CD80 or CD86, and it gets expressed on on the surface. Where did the cursor go? Yes, it gets expressed on the surface. So this one. Well, along with that, it does another thing is it gets activated and releases certain cytokines. And this cytokines, interleukin 1, 6, 12, 4, any kind of cytokines. Now, what type of cytokines this antigen presenting cell will release, it actually decides is the differentiation of the naive T cell into that particular phenotype of the T helper cells. Now, what this antigen presenting cell does, it then goes from the area where there's inflammation to the draining regional lymph node. And it activates as the quiescent to the naive T cell over there. So now comes the role of the activation of the adaptive immune system. So here then this is the naive T cell. It gives a signal 1 where the T cell receptor it binds with the MHC class 2 or the class 1 and the peptide number 1. Signal 2, which is given by the CD28, which is a co-stimulatory molecule binding to a CD8086, and obviously signal 3, where the cytokine, it binds to the cytokine receptors. So when all these three signals, signal 1, 2, and 3 goes to a particular naive T cell, this T cell gets activated, and it leads on to the clonal selection of that particular T cell, and a clonal expansion and differentiation. So what happens is that, so now we discuss about come to the adaptive part of the immune system. So let us in short go through, recapitulate or try to revise quickly about what components of the adaptive immune system. See, the adaptive immune system, they are actually, it has two components again. One is cellular component, another is the humoral component. So cellular component is mainly, it is provided by the T lymphocytes, and T lymphocytes, they are called the king cells, which actually orchestrate the entire immune function, and also it provides a help to the fighting innate immune cells else in the border area, that is in the area where this infection is. And so another component is humoral immune system, which is mainly provided by the antibodies, and antibodies are basically secreted by the B lymphocytes or the B cells. Again, these B cells need a help from the T cells to actually activate and produce and release the antibodies. So let's talk about the cellular immune response or the T cell function first. Okay, so what happens is that now this antigen presenting cell has gone into the draining regional lymph nodes, the secondary lymphoid organs, and it has activated the naive T cell by providing three signals, signal 1, 2, and 3, as we have discussed. Now, once this naive T cell has got activated, so if it is an MHC class 2, then this naive T cell becomes a T helper cells. Okay. Now, depending upon what type of the cytokine signal it gets, it can become either a Th1 or a Th2 or a Th17 or a TREG or a Th9 cells. So if the cytokine signal is interleukin 12, then the naive T cell becomes a T helper 1. The cytokine signal is interleukin 4, then the naive T cell becomes and Th2 cells. If the cytokine signal is the TGA beta or interleukin 10, then the naive T cell becomes a TREG cell. Okay. So this is how it happens. So once this naive T cell has become a T helper cell, and that too of a particular phenotype, then this T helper cell, what it does is that it releases its signature cytokines. And releasing the signature cytokines, it again goes back to the site from where the APC has come, and then it goes into that area, and it gives that help to the fighting innate cells over there. Okay, so if it is a Th1, it will go to that site, it will release its signature cytokine, and that is the interferon, that is a gamma interferons, and it will help the macrophages over there to ingest and kill the organism. So it helps in intracellular killing and by the macrophages. So if it is a Th2, so Th2, what it will do is that it will go to the site, it will release its signature cytokines, that is the interleukin 4, 5, and 13, and it will give help it will cause the accumulation, survival, activation of the eosinophils, else and at the same time it will cause the activation and of the eosinophils, and also it will help and the function of the eosinophils to release the various granules and they destroy the organisms over there. So if it is a like Th17 cell will release its signature cytokines, namely interleukin 17, interleukin 22, which helps in the killing of the extracellular organisms. And which is a Treg cell, then Treg is basically important in suppressing the exaggerated immune response. So this is how this happens. And or if it is an MHC class 1, then the MHC class 1 will cause us the activation of the naive T cell to a cytotoxic T lymphocytes, or the CTLs. And the cytotoxic T lymphocytes, it will go to the particular site, and there it will release is the various cytotoxic granules, maybe like the perforins gran and enzymes, and will cause a destruction of the cells or the organisms in that a particular area. So how the T cells are formed? T cells are basically, they are they they first of all they come from the bone marrow, but they develop in the thymus. Okay, so how it develops, like I'm just in brief, I'm telling you what happens is that the common lymphoid progenitor cell, it arrives at the junction of the cortex and the medulla, and then it enters the cortex. Okay, so in the cortex, it goes through a series of the changes. As like initially it goes into a double negative phase 1, DN1, DN2, DN3, and DN4, and then it forms the double positive cell. So what happens during this maturation phase in the cortex? During this maturation phase in the cortex, the T cell receptors develop. Initially, it is the beta arm, then the alpha arm, so it develops, and then what happens is that the double positive stage, it has both the CD4 and the CD8 molecule. So at the same time, various mechanisms occur in the cortex. One is called the tolerance, central tolerance mechanisms, or here one thing happens is that the positive selection. What is meant by positive selection? So there are certain cells in the cortex which are called the cortical thymic epithelial cells, or the CTEC. What the CTEX does is the CTEX then actually it shows the various self antigens to the to this developing T cells in the cortex. So once these double positive cells are formed, okay, so if these cells can recognize either the MHC self MHC class 1 or the self MHC class 2, which is shown by the CTEX, those cells survive. Okay, if those cells cannot recognize any of the self MHC class 1, self MHC class 2, then this interaction between the self MHC class 1, class 2 with the T cell receptors, it gives them certain surviving signals to the cells. If those are not there, these cells die by neglect. So means those cells which can actually recognize this, they get positive, they get selected. So this is called a positive selection. So once these double positive cells they are formed, they goes into the medulla. Okay, and medulla, what happens? Medulla, there's another interesting cell which is called the medullary thymic epithelial cells, MTEX. What is MTEX? They has a very unique kind of gene in them, which is called the IR gene, also known as the autoimmune regulator gene. And because of this autoimmune regulator gene, what this MTEX does is MTEX produces various tissue-specific antigens and also called tissue-restricted antigens, TRAs, and produce it on their surfaces. At the same time, in the medulla, various as antigen-presenting cells, they bring the various self antigens to to actually the developing T cells. So what happens is that if the T cells, they recognize very strongly the self self-antigens, they usually die by apoptosis. It is a very strong recognition. So this is called, and those cells which do not recognize the self-antigens, they survive. This is called a negative selection. Okay, and those cells which weakly recognize as or with a moderate affinity, they recognize the self antigens. Usually these cells, they become a regulatory T cells or TREGs. So this is how the thymus ensures that the T cells which is being released into the circulation, they do not react to the self cells, but these cells can actually recognize only the foreign antigens and develop an immune response against that. And so this is called a negative selection. So after all the selection processes, positive and negative, the T cells are ultimately released into the circulation. Now, I think I forgot to tell you that is this double positive cells, else they usually become a single positive cells in the medulla. How? Because there are two, the CD4 and the CD8. So you know CD4 recognizes MHC class 2 and CD8 recognizes MHC class 1. So if there is initially this cell by chance comes in contact with the MHC class 2, then there is a stronger and longer interaction leads to the and there is transcription factor LCK. There is a LCK activation, and which leads on to the downregulation of the CD8 expression. This cell becomes a CD4 positive. It comes in contact with the MHC class 1. There is a shorter and weaker interaction with the LCK. It leads into the downregulation of the CD4, and this cell becomes a CD8 positive T cells. So we have mainly two pools of the T cells, CD4 and the CD8. And the CD4 positive naive T cells goes on to produce the T helper cells, and CD8 positive T cells goes on to produce the cytotoxic T lymphocytes, as we had discussed. So this is in short about the development of the T cells in the thymus. So these T cells not only orchestrate the functions of the innate immune system, but these T cells also provide the required help to the B cells to produce the antibodies. How we'll just discuss within the next 2 or 3 slides. And these T cells release this helper T cell release the specific cytokines and produces the cytokine milieu. In that particular milieu, it leads on to basically that particular immune response, which is desired and which is directed by that particular pathogen. We just discussed about the CD4 and the CD8 positive T cells. So there is a very interesting synchrony in the entire nature. That is how this CD4 and the CD8 cells are developed. There are two major. There is this antigen presentation, which is called as a unique antigen presentation. Let me just briefly discuss it with you. You see, there are two major pathways of the antigen presentation. One is a proteasomal pathway, and another is a lysosomal pathway. Now, what does this lysosomes does? So lysosomes, this is basically, it is formed. So whenever a innate immune cell, like antigen presenting cells, or a macrophage, which is also an antigen presenting cells, or neutrophils, or whatever it is, so it comes in contact with an extracellular organisms. It engulfs that organisms. Once that organism is engulfed, so then and it forms a vesicle. Okay, and this vesicle, it fuses with the lysosome, and the lysosomal enzymes, it degrades that at organisms. So this degradative compartment which is formed is basically, it is linked to the external environment. Now what happens is that this is lysosomes, it then fuses with the vesicle which is formed from the Golgi apparatus, the Golgi, which has the MHC class 2 proteins. It goes and takes up that peptides, and again that compartment, and it fuses with the cell membrane. So what happens is that MHC class 2 is basically, it is taking up the components, or this degradative compartment is formed, which actually takes up the external microorganism and juts it outside the cell membrane, which is recognized by the T helper cells. So this is basically the lysosomal compartment, or lysosomal pathway antigen presentation is basically for the extracellular organisms. Now there is another pathway which is called a proteasomal pathway. Now what happens in a proteasomal pathway? We know that in a cell, so there are the various organelles in the cell, and the cell has a particular lifespan. So what happens is that those proteins which are present in the cytoplasm, which is not connected with the outside of the cell. So these proteins, they are actually degraded by another organelle in the cell, which is called a proteosomes. So this proteosomes is destroys as the proteins which are present in the cytoplasms, and this peptide are then transported into the endoplasmic reticulum through a protein called the transporter protein, and it then binds to the MHC class 1 over there. And this MHC class 1, when binds to this the protein peptides which degraded by the proteosomes, it juts out of the membrane, and which is being recognized by the CD8 positive T cells, which forms as CCTLs. So this can actually destroy these cells, which is jutting out the degradative proteins from the internal compartment. So showing that this cell needs to be killed. Okay, so that is how it is being done. So these are two ways of the antigen presentation. One is proteasomal pathway, which is linked to the CD8, and there is another pathway, that is the lysosomal pathway, which is related to the CD4. Interestingly, if you see the viruses, so the viruses actually, they go and embed itself into the that is the genes ends of the host cell, and it forms using the machinery of the host cells. It forms the viral proteins which comes into the cytoplasm of that host. So what happens is that those proteins can be degraded by the proteasomal pathway, and with the MHC class 1, they juts out and then recognize CD8. So though virus is an extracellular organism, which is outside, it is in the outside environment, it uses the proteasomal pathway for the antigen presentation. This is called a cross-present presentation, which is basically required for the killing of the viral infected cells. So we see that any cell-mediated immune response has two components. One is a primary response, and other is a secondary response. Now, what is the primary response? So once the naive T cells has seen and or come in contact with the APC, which is showing that particular peptide along with all the three signals that we've just discussed about, it goes and starts killing. At the same time, there is a particular type of effector T cells which is formed from the CD4 positive T cell, which is called T follicular helper cells. And these T follicular helper cells are basically needed to activate the B cells. So there is also a T cell and a B cell interaction, which occurs at the junction of the cortex and the medulla in the lymph nodes. And these B cells, when they get activated, they move into the cortex, into the follicles where they form the germinal centers. And again, with the help of the T follicular helper cells, they actually become activated and start. They form the plasma cells, and these plasma cells can actually produce the antibodies. At the same time, these B cells also form a small clone of the memory B cells. This memory B cells, it sits quietly in the secondary lymphoid organs, or maybe in the gut, or maybe in the bone or marrow. And in the second time, when this memory B cells are actually already, they have recognized as they are produced again the same infective organisms. And this memory B cells, for its activation, it do not require again another help from the T cells. It can activate very fast. Okay. So when this memory B cells, it is also an antigen presenting cells. When it comes in contact with that particular infectious agent again, and it comes and binds to the B cell receptors on the memory B cells, it gets activated without a T cell help. and this forms a clone on which can rapidly it can transform into the effector B cells, and the B cells into the plasma cells and release the antibody. So what happens is that there is a delay, so of few days, or 4 to 5 to 7 days, from when the infectious organism enters the body for the first time till there is the initiation of the adaptive immune response. And this is called the primary response. So the adaptive immune response, which happens for the first time, it is slower in onset, number one, and it is mainly a form given by the IgM and to certain extent IgG, but as and the titer of the antibodies is also low, and affinity of the antibodies towards the organism is also lower. So what happens is that when it comes again for the second time, there happens a rapid response. It that does not limits to after a few days. It happens immediately. Number one. Secondly, the titer of the antibody is also much higher, and the antibodies are much affine. So there is affinity maturation of the antibodies which takes place in the germinal center, and there is also a class switch combination. So the higher affine and more effective antibodies of the IgG a class is produced in the secondary immune response. And so when we this is means we had just talked about the how this response initiated. So when we say that for the humoral immune response to be initiated, first again, and see and a protein is a big structure. Okay, it has as many peptides into a three dimensional conformation. So what happens is that some of the peptides, they are into the beta pleated sheets or in the alpha helices. So these are the primary structures in a protein. So what happens is that all the peptides which is inside the protein, they which are in a single straight line, they are called the linear peptides. And if they're recognized by an antigen presenting cell through its receptor, it is called epitopes. So this could be linear epitopes which are present inside the cell, which are actually when the cells, which are inside the protein, and when the protein is cleaved, these linear epitopes are presented by the MHC class 1 or 2 to the T cells. So the linear epitopes are basically for the T cells. But when it is a three-dimensional structure, the peptides present on the surfaces, they are recognized as by the B cell receptors. B cell receptors are actually the membrane-bound IgM or IgD in the B cells. So these are called the conformational epitopes. And so a single protein, it may have some peptides sticking on the surface, some peptides deep inside. And when this protein is cleaved, so they have two different sets of the peptides. So one of them is recognized by a T cell, another of them is recognized by the B cell, but both, because of the synergy because of the same protein and the same antigen presenting cells, they're actually cleaving them. So it is interesting that there is a coordination between that B cell and that T cell, though they are seeing two different parts of that same protein. This is called the T cell B cell coordination. So what happens is that these B cells, with the use of the chemokines, which is basically needed to come into. So there's a unique apartheid between the T cells remain in the T cell zone as a parafollicular zone, and the B cells remain in the B cell zone in the lymph node, that is the follicles. So they don't intermingle with each other. So only when there is this particular this antigen peptides are broken down, these B cells, then they start having receptors for the T cells on chemokine CXCR7, and it comes in the T cell zone. And there is a particular molecule which is expressed on the CD40, and there's a CD40 ligand, and they both come attaches with them along with that, along with the MHC class 2 and the peptide. And this actually T cell then wax up. The B cell causes its activation, and then forms the short-living plasma blasts, and the plasma cells are release of the IgM. So at the same time, what happens is that this T follicular helper cells, which develops from the naive T cells, it along with that, the B cells, which is activated, it goes into the germinal center, and then it gets activated, and it start forming the antibodies against that particular conformational epitope which it had seen. So initial the antibodies which is produced is of the IgM, but there happens two important steps in the B cell and germinal center reaction. One is the class switch combination, where it happens that though the antigen binding site remains the same, but the constant region changes from the IgM to the IgG, okay, or maybe IgE or maybe IgA. So, and this is needed for because of the type of the organisms it is encountering. Because if it is a mucosal some antigens which is entering to the gut and all, it needs IgA. It will produce the IgA. If it is say a parasite, it needs the IgE. It will produce the IgE. And if it is any other organism, it needs IgG, because IgG is the most effective antibodies. At the same time, with every cycle, there happens some maturation and of the antibody binding site, so that this antibody becomes more and more effective in binding to that antigen. This is called affinity maturation. So these two things is mediated by that is a type of the dendritic cells which is present in the follicle, which are called the follicular dendritic cells, and also by the T follicular helper cells. So now these antibodies which are formed, they may performs few of the major functions. What are the functions of the antibodies? So the functions of the antibodies, first is the neutralization. What is the neutralization? That is, it can bind and neutralize any foreign an antigen or an organisms, which is mediated by IgM, mediated by the IgG. The second component is opsonization. That is, it can bind to a microorganisms and make it more palatable to be engulfed and digested by a phagocytic cells. So this is called opsonization, because an IgG is very important for the opsonization. It binds to organisms, and because of the phagocytic cells has the receptor for the constant region of the IgG, mainly Fc gamma R1. So it can bind to the IgG, which is bound to the microorganisms and engulf and digest it. And that is opsonization. Second is that it is the antibody dependent cell mediated cytotoxicity. What is that? This antibodies binds to an organism, and through its Fc, if that constant region, it can bind to the high affinity IgG receptors on the natural killer cells. NK cells, when it gets bind through the high affinity IgG receptors, it can basically get activated, release the perforin enzymes, and kill that at organisms or kill that cell. This is called the antibody-dependent cell-mediated cytotoxicity. Fourth is that it can do a complement activation. And this ADCC is mainly mediated by IgG. Complement activation, that is, we have seen before, it is by the classical pathway, by the antigen-antibody complexes. It can activate the C1q, and then C1q activation can lead on to the classical pathway activation and C3 cleavage. This is mediated again by IgM and IgG. Okay, and another important is the mucosal barrier immunity. This is provided by the IgA, and another is a mast cell activation, which is being performed by the IgE. So, but we see that the neutralization, the opsonization, the complement activation, and antibody dependence limited cytotoxicity, these four are the main mechanisms how the antibodies is help in killing an organism. And along with that, definitely that mast cell activation and the tissue immunity is also part of this by Ig and IgE respectively. And out of this, only IgG can cross the placenta. So that is one part of that. So antibody structure, well, they basically belong to the gamma globulin fraction of the serum proteins. They have two identical heavy chains, two identical light chains. There are five kinds of antibodies: IgG, M, A, D, and E. Uh, and the IgM is the first immunoglobulin which is expressed by the B cell. It causes opsonization. It causes complement fixation. It also causes neutralization. IgG neutralization, opsonization, complement fixation, and it only can cross the placental barrier. IgD is important. IgD here it's written unclear, but IgD is important in actually B cell maturation, and also it plays an important role in the B cell tolerance, and so it has an important role in that. And IgE, it is important immune parasites, and it is also causes allergic reactions. And IgA, the mucosal surfaces, produce mucosal toxins, bacteria, viruses, and by binding and then neutralizing the various organisms in the mucosal surface. So we have talked about the sequential IgM, IgG humoral response in the primary and the secondary. This is the primary and the secondary. So there's certain immune effector mechanisms. Always this I find this table to be very, very interesting. Like if it's extracellular bacteria, so immune system protects against four classes: extracellular bacteria, parasites and fungi. It could be intracellular bacteria and parasites. It could be viruses. It could be a parasitic worms. These are the main microorganisms. So for the extracellular, we have, that is the innate immune effector mechanism, type 1, type 2, type 3. Similarly, we have the adaptive immune response. Also similarly, that is, we have the type 1 immune response by Th1, type 2 by Th2, and the type 3 by the Th17 cells. So this we have talked about, that the neutralization, opsonization, complement activation, ADCC, and all the effective mechanisms. So if we go into summary, innate immunity relies on mechanisms already existing before microbe infects host. The first line of defense has the memory, but nowadays we talk about the trained end and memory, or that is the TRIM, or the trained and immunity. So this is this is a new concept which has come up as a memory for the innate immune system. Adaptive, it follows after development of the microbes. So it is adaptation of the body after it comes in contact with basically microorganisms. So it's very adaptive. So it only if the innate fails to clear a microorganism, it seeks the help of the adaptive immune system, and which strengthens its arms. It has specific components of T cells and the B cells. We talked about the primary and the secondary immune responses. And a failure immune response can lead to an immune deficiency or the hypersensitivity. So if infectious agent and effective response to an antigen is normal, is protected, if it's deficient, we get recurrent infection. If the innocuous substance, if there's a body is reacting to it, it's a normal response is allergy. It should not react to it. Deficient is no response. It is what is needed. Grafted organ, if it is reacts to it, immune system, it can rejected. Deficient response is accepted. Self organs is reacts to it, it's autoimmunity, and deficient is tolerance. Tumor is normal response, tumor immunity, but if it's deficient response, tumor cells leads on to the formation of the cancer. So, so thank you very much. This is the CMC Ranipet campus, and I'm an adjunct faculty over there. And basically, I love teaching immunology to my students there. Anyway, so I stop here, and I would be very happy to take up questions, any if it comes.
Any aspirants, if you have any question, you may ask now.
Anything. If there is no question, I won't be very sure that whether I was able to communicate the entire thing or not. Anything, any question regarding immunology? I've discussed in nutshell about the various parts and the functions of the immune system.
Sir, I believe the aspirants don't have any question as of now. Sir, shall we schedule a class just for question and answers? The A type and K type questions.
Okay, so what you can do is that you can just collate the questions once they have it, and then we can schedule a class, okay, regarding the questions, okay?
Sure, sure.
Okay, okay. Thank you very much.